Okay, so this is the next chapter. So how are we going so far? Am I talking very fast? Paspas lang adoro, ana. Miss Puritana.
Okay man lang po, Dok.
Nakalanat ka
Okay lang
mo?
po, Doc.
Opo, nakakalanat pa.
Okay. Yun ko. Amo itong takon kuan. Amo itong takon speed. I can maluya pa nga nito. Kaya wala ko pamahaw. Kaya ko pamahaw fast ang speed. Oo. Kaya sila ito rin magluya ha. Although I'm getting old, akong dila paspasay lagi ha. Joke lang ito ha. If niyo pag-awaya, kaya sa kapala ka istorya, 20 na itak. Di ka madaog ha. Amo ito nagpanhadlok. Nako ay, salit ko, wala yung away na ako. Wala na away ako. Kaya sa pala akong istorya, o sa pala tira, 20 na itak. Nagbura-bura na ba. Tapos, pag di ko na mailog, magpaspas ka pa kamot. Madali ka pa, manaplong, suntok pa. Salit pa rin nakikaway ako. I remember that since I was a kid. Ganon. So, kung mo pa ngayon ang tunga trait, nag-develop ko as I get older. Joke. Oo. Like, okay nga nito nga trait nga develop. I remember when I was a kid in my hometown. So, wala mo rin mananak mga kwa. Di rin nahira nakikaway. Masa, maano na nga niya ako. When I get angry, nahupoy na nga tanan.
Nahadlok.
Hadlok na yun. Siguro tungod niya paspasay, ulit nagyayakan. Kumakapas. Amo to takon, that's why I kept asking the student, am I going too fast? Because I tend to talk very fast. Kung naghihinay, bakit yung pipiraw malatak? So, yan na. If I'm so, paspas la, you just tell me, I can slow down.
Sige po, Doc.
Thank you. Okay, so next, we'll go into immunity. Immunity na tayo. So there are two types or two categories of immunity. So one is the non-specific and the other is specific. So you look at this, wala mo ng pula siya. May balik ang pula. Di mas na-klaro ka iya. Okay. Okay, thank you. Pasensya na. That's also the drawback. Kalagas na. Hindi na kanya masyadong maaram ng mga kuwan. Masigod ka, matindigod pangangailangan ni Doc. Bislagas na. Ganyan na extra pa. I wanted to quit na. I retired from government service 8 years ago. Pero di man ako makaka-retire pa din. RTR. Makarawad sa pagbaya. I resigned from Southwestern just this year. So, di na ko kay kapoy na ganin niya ay di ko kapas ng technology, everything online. I have internet connection at home, but para ito pag para messenger, tawag ako mga siblings abroad, mga friend, mga classmates, pero mga connecting like this, I didn't maram. Okay, so we'll just continue. Bear with me. So non-specific defense and specific. So duha ka klase ang aton immunity or immune system. This is how it works. So the innate immunity or non-specific, it will protect us against foreign substances or cells without having to recognize their specific identities. So the mechanism of protection used are not quite unique to that particular foreign substance or a cell. Like for example, you inhaled an organism. So maagit het irong, di ba? It passes through your nose, your uh, external nose, then into your So as it passes through that, di ba, may mga buhok-buhok aton the what do you call that, an anatomy, the nasal canal. Di ba, ciliated doon dida. Labutan the microscopic cilia, di ba, may mga buhok-buhok dida sa kubi mo irong. This is part of our natural defense. So it will try to trap whatever foreign substance or foreign antigen that we inhale. Likewise in the mouth, kakaon ka something, you eat something, and it's not good. May bacteria, it's infected, or contaminated food or water. May mga enzymes in the mouth that will try to kill them. So natural. So non-specific or non-specific. Well, specific immunity is also called acquired immunity. So it depends upon specific recognition by the lymphocytes. So remember, it's only the lymphocytes of the substances or the cell to be attacked. Why the lymphocyte? Because these are the only cells in the body that are immunologic. They have the capacity. The other cells, mga non-specific to the other WBC. Ito lang. And this is followed as upon recognition that by that particular lymphocyte of that attacking antigen, followed by an attack that is just unique for that particular substance of the cell. So that is why it's called specific. Okay. So innate immunity or the non-specific. So it includes phagocytosis of bacteria and other invaders by WBC and tissue macrophages. So we learned that already, how a phagocytic cell works. So the bacteria is engulfed and then it forms a phagolysosome fusion, and then inside that phagolysosome tries to kill or break down that bacteria. Okay? So again, another way is destruction of the swallowed organism by the acid secretion in the stomach and by the digestive enzymes. So this is the reason why you went for a picnic, kaong kinilaw nga tangigi. Okay? But the tinilaw has been there for quite some time. Then it becomes contaminated. Or a classic example we give in micro exam, the student at a party, nangaon spaghetti or salad, etc. Okay. Then some of you will come down with diarrhea and vomiting, but others not. Okay. So katong di nag-diarrhea or mag-vomit, they have their innate immunity was working. Like in cholera, if you consume a food with Vibrio cholerae, why is it that you don't come down with massive diarrhea, watery diarrhea? Because cholera organism that you swallowed and then it goes into your stomach, they're destroyed by the stomach acid. Unless you have an overwhelming amount of Vibrio cholerae in that food that you consume, nga di na na-contain ng gastric acid, so yun, you come down with cholera. But there are organisms also that can survive in the stomach acid, like Helicobacter. Okay? Then resistance of skin to invasion by organism. So in micro, that's the first thing we tell the student. An intact skin is a good protection, a barrier against infection. So aside from an intact skin, the skin has certain secretions, okay, that are detrimental or not suitable for an organism to grow. So resistance. Like another is the presence of a normal flora. Okay, so we know that our skin, it's not sterile. Okay, maski magsinabon kapag safeguard, di ba? the patalastas ng safeguard that it kills 99.9% of germs. So may nabilin nga 0.1. So the 0.1 multiply. Okay, generation time is 20 minutes. So sige multiply yung bacteria. So it does not kill the organism. After 20 minutes, mag-regrow, nanapod matubo nga bacteria. Okay, so the skin has a natural way of warding off microorganism. Mga secretions in the skin, fatty secretions, acidic secretions in the skin. And then presence of in the blood of chemical compounds that attach to foreign organisms or toxin and try to destroy them. So these are natural or non-specific mechanism. Okay. Before that, the physiologic mechanism, like sneezing. Okay, sneezing is a physiologic mechanism. So maybe you inhale something, diba? Foreign, diba? Kaon ka na suripan. What happens is you try to cough it out or sneeze out. Nanggawas ang mumho. Then, so it's a physiologic mechanism. Crying, sample ko ito nga, crying. So, when you fail physio, fail ka sa quiz, fail ka sa bi-monthly, hilak ka, cry. Or you watch Korean telenovela, medyo sad, sige ka, hilak, cry, sige, hilak. It's all right. Hilak lang. The tears has lysozyme. So it can wash away the microorganism. Because lysozymes are good against, especially against gram-positive bacteria. It can melt the peptidoglycan layer of your bacteria. So, if you failed in one quiz in physio, sa micro na, example na, failed ka sa micro quiz, just cry. But I pag-cry of the whole week, the whole day, cry for a few seconds. Siguro, then, you reassess yourself. Maybe I need to study more, read more. Di rin alam po niya, sige, cry. It's no longer physiologic. Baka kato ka na. So, it's all right to cry. Because if your mother scold you, cry. But do not cry the whole hour. Just to let go, let that steam out. Okay. Ako, I cry. Makita ako ng Judy Ann Santos, yung mga drama. Pag makatarangis, I cry. Then my children say, "Oh, Nay, are you crying? Why are you crying?" I am just washing my eyes. Okay lang yun. Okay. So, yun. Those are not physiologic mechanism. Kanang mag-uro-uro ka, diarrhea, it's physiologic. That's why we do not prescribe nowadays anti-diarrheal. We just let the pass out those liquid stools until all the toxins are out. Because if you give anti-diarrheal, you just stop the diarrhea because it's antispasmodic, stop the movement of the intestine, the motility of the intestine. What happens? So the bacteria are still there and continue producing the toxin, and then you continue after a few while. Balik diarrhea. Okay? So those are physiologic process. Coughing, sneezing, diarrhea, crying. Okay lang yun. Pero ayun nga, like say, when the diarrhea has been prolonged for some time, na-dehydrate na ka, nanlaw na yung mata, it's no longer physiologic. You need to go and see a healthcare provider. Maybe you need to be hydrated. Para ng cholera, it's a massive diarrhea. It's no longer physiologic. Sobrahan. Okay, so these are the example pala on yun. Lysozyme, it's a mucolytic polysaccharide that attacks bacteria and causes them to dissolve, especially the cell wall of the bacteria, the peptidoglycan layer in gram-positive. Okay, basic polypeptides which react and inactivate certain types of gram-positive bacteria. Then the complement complex, this will be discussed later. It's a non-specific, a system of about 20 proteins that can activate in various ways to destroy bacteria. Then natural killer lymphocytes or NK cells can recognize, destroy foreign cells, tumor, and even some infected cells. Okay. So the next one are the adaptive or your acquired. So what happens in the acquired immunity is you form antibodies or activated lymphocyte. Diba? We said there are two populations, the B cells or B lymphocytes and the T lymphocytes. So the B lymphocytes, these are the ones that forms the antibodies, and then the T lymphocytes, they are activated and they will attack and destroy a specific invading organism or toxin. So they mainly involve the lymphocyte. So again, the lymphocytes are the only immunocompetent cell in the WBC series. Remember that, Permitnaga was a tiksam. Ano siya? Okay, so what happens, these are initiated by an antigen. So an antigen has to enter the body. So what are the properties of an antigen? So antigen are proteins, usually proteins, but it's not only a monopoly of proteins to be antigenic. So mostly proteins or large polysaccharide molecules, they are antigenic. Then they have to be of high molecular weight. So 8,000 or greater than 8,000 kilodaltons will be the molecular weight. Because if they are small, they are very weak antigen, and then sometimes they cannot stimulate the adaptive immunity to initiate that response. But there are instances wherein low molecular weight substances can become antigenic. So example in the textbook, permito in example namon, penicillin. Okay, penicillin is a low molecular weight protein. But how is it? A lot of people get allergy to penicillin. Oh, damo, you will hear, di ba? Usually, pag na-doktor ka na, or residente, or a clerk, yeah, in taking history, any allergies, ma'am, to food, to drugs? So a lot of people get allergy to penicillin. So why? If it, come on, may low molecular weight of penicillin. But what happens is the penicillin substance, it will, coupled with a tissue protein in our tissue, and it becomes a bigger molecule. Now it becomes antigenic. So ito, amot reason for certain persons. Okay. So the penicillin plus the tissue protein of that individual, so it becomes, and then the antigen will have an epitope or the antigen binding site. So these are regularly recurring molecular groups found on the surface of a large molecule, and that's the identifying mark of the antigen. So it must have epitope or the antigen binding site, so for that to bind, to be able to attach. Okay. So the two types of acquired immunity could be humoral immunity or the B cell immunity or the antibody mediated immunity, and the cell mediated or the T cell immunity. So in humoral, the lymphocytes are involved. So you have B, that's why it's called humoral or B cell immunity. And what is produced or what is used as an antigen killing apparatus here are the antibodies that are produced by an activated B lymphocyte. So upon antigenic stimulation, the activated B lymphocyte, they differentiate into a plasma cell, and the plasma cell are the ones that will produce this. Okay. While in the cell-mediated, it's the T lymphocytes that are involved. Okay. So there are no antibodies formed in cell-mediated immunities. So cell act directly on that invading antigen. Or the T cells are, okay, so the T cells are divided into three subsets. So you have a cytotoxic T, a helper T, and a suppressor T. Okay, so cytotoxic T, I discussed that in the preceding chapter, and the helper T. Then the suppressor T or regulator T cells. Because what happens, sometimes you get an overwhelming or an overacting T system. So, what will happen is, kung nasubrahan, that's why we have this so-called autoimmune disorder. Okay? So, that's a function of the regulatory cells. Yeah, oy, you're overacting, tama na. Yan. So, that's how this one function. Okay. So, next, this is on, and this is a cross-section of a lymph node. Okay. And so we have millions of specific lymphocytes are stored in the lymphoid tissue. And then its preformed lymphocyte is capable of only one type of antibody or only one type of T cell with a single type specificity. Okay. And then only the specific type of antigen in which it can react and can activate that particular T or B lymphocyte. And once they're activated, it will reproduce, forming tremendous number of duplicate lymphocytes that act only against the particular antigen. So where do lymphocytes come from? How to differentiate? Okay. So again, medyo magulo ini nga slide, but we will just walk through this. This is para maintindihan. And once you understand this slide, it's easier for us to understand everything. So immunity and allergies. B anti-lymphocytes are derived from the pluripotent hematopoietic stem cell in the embryo, di ba? They are from the lymphoblastic series. So you have your T lymphocytes. What happened is they migrate first and they are pre-processed in the thymus. That's why they're called T. And shortly before birth and for a few months after birth. So they are the immature T, they are pre-processed in the thymus, then they mature here in the thymus. And they are the ones responsible for your T cell or cell-mediated immunity. So upon maturation, you will have a mature helper T. Okay? While the B lymphocytes, they are pre-processed in the liver during the mid-fetal life and then late fetal life, processed in the bone marrow, and they are the ones that are destined to form your B cells. So this one is from the lymphoblast, from the pluripotent hematopoietic stem cell. So we have the lymphoblastic cell giving rise to the T and the B. So di ba may usa pa ini an NK. But we will not include the NK because we're discussing the specific immune system. Okay. So your mature B cell, it goes into, from the bone marrow, goes into your secondary lymphoid organs. So upon antigenic stimulation, what happens? So your B cell will proliferate, differentiate, and it will transform into a plasma cell. Okay. So the plasma cell now will produce antibodies against that particular invading antigen. Okay? And then, ang T cell naman, same, it matures in the thymus, and then it becomes a mature helper T and a mature cytotoxic T. Then upon antigenic stimulation, so an antigen stimulates to, it will stimulate to proliferate more cytotoxic and more helper T cells. Okay. So mechanism of activation of a clone of lymphocytes. Its B cell has on its surface, so this is a B cell, has on its surface about 1,000 antibody molecules that will react highly specifically with only one type of a specific antigen. So this is, This is the B cell, and you look at the red one, red Y-shaped structure. These are the immunoglobulins, and this is the upper portion. This one here, it's the variable portion where the antigens are attached. So for attachment of the antigen. So upon activation by an antigen, what will happen? Your B cells will be cloned to produce more. So madaming B cells, and the B cells differentiate or will transform into a plasma cell. So the plasma cell will produce antibody. Then from the clones of B cell that are produced upon stimulation by this particular antigen, some does not form into plasma, but some becomes memory cell. Again, memory cell. That is why only the lymphocytes are capable of, that's why they are called immunocompetent, because they are the only cell that are capable of immunologic memory. Selected my memory. According to my son, nakainom siguro na yung memory gold class. Siguro. So yun. So you will have a population of memory B cell that will be stored, and this memory B cell in particular specific siya for these antigens. Example, antigen X ni. So the memory cell is for antigen X. So upon second encounter with antigen X, kining a memory B cell that has recognized this one will be immediately activated. So kumbaga, secondary response na. Okay? So encounter with the antigen stimulate the lymphocyte to make copies of itself, and the clones divide into two. So one group becomes plasma, forming antibodies, and one group is kept and reserved as memory cells. Okay, that's it. For T lymphocyte, same. Its cell has a molecule similar to antibodies called as surface receptor protein, or your T cell markers. So on its membrane, these two are highly specific for one specified activating antigen. Then the antigen binding also activates T cells, but it differs from that of the B cell. So this is a helper T. So upon antigenic stimulation, you have a clone of helper T being produced, and some of them becomes memory. So yun. So it's in the thymos, I said it's in the thymos maturation. So a mature T cell upon antigenic stimulation, yun. You have this helper and then a mature cytotoxic, two groups. So again, you will also have memory cell, memory cytotoxic, and a memory T helper. So activation of B cells and other factors needed. So although the macrophage is not part of the specific defense, it helps, no, in the defense system of the body. It's not that when a foreign antigen comes in, non-specific ka mula anay, mapahuway lanay kami specific. It's not. So there is some sort of a cooperation between these two. Dirito pareho dida aton group kamula ni trabaho dida kinita lanay kami. Hindi mo na kaya, just call me. It's not. So it's there is a cooperation. So your macrophage, when it phagocytizes an antigen, it will present that particular antigen to a B cell. And at the same time, that particular antigen is also presented to a T cell. So you activate the helper T cells are formed. Okay? Then some B cell clones become plasma, and the other becomes memory, diba? And secretes antibodies specific for that antigen. Then the other becomes memory cells. So pareho gap din with so your helper T, you will have high cytotoxic. So upon antigenic stimulation, you have a clone of cytotoxic and a clone of helper T, and then your cytotoxic or directly attacks antigen-bearing cells, and then at the same time you will have memory cell formation. So two types of antibody formation. So we have the primary response and the secondary response. So the primary response is your first contact with an antigen. Uh, the body forms antibodies. So the process takes lower, a 1 week delay in appearance, and then the antibody form is of weak potency. Usually it's the IgM that rises first during the primary response, and it has a short life, so shorter lifespan. While a secondary response, the red one, if you look at this, it occurs after secondary exposure to the same antigen because of your memory cell, that the memory cell recognizes that same antigen. So second time exposed to the same antigen, it begins to rise rapidly within hours. While it takes about a week for your primary response to pick up an antibody, but in secondary response in a few hours, you will have antibody in the system. And it's more potent, and it forms antibodies for many months rather than for only a few weeks. So you're in the primary response, it's just a short life, sorry. So in the secondary response, the antibodies will stay in the system for months, okay? And then this is attributed to activation of the memory cells. That's why you have to understand how even the function of the memory cell. So increased potency and duration of the secondary response. This is the rationale in immunization. Remember, immunization we give like DPT, we give 3 doses. So to enhance uh, memory cell formation, activation of your memory cell, and then we give booster shots, okay, so that you will have more antibodies produced in our system. Okay, so what are antibodies? Nature. So these are immunoglobulins. Gamma globulins are called immunoglobulins, and this is, you will get to see this abbreviation. So IG, capital I and a G, that stands for immunoglobulin. And then depending on the type of immunoglobulin, so you add the A, if it's immunoglobulin A, immunoglobulin M, IgM, IgG, IgD. So there are 5 types, okay? So their molecular weight ranges from 160,000 to 170,000, depending on the molecule, because IgM is the biggest and the heaviest of the antibody molecule because it's a pentameric compound. So this is a monomeric compound of an immunoglobulin molecule. So kung pentameric for IgM, so that it has 5 like this, 5, 5, 5 like this, 5 Y-shaped structure. And usually, they constitute 20% of all plasma protein and antibodies. Then immunoglobulins are made up of a light and a heavy chain. So if it's clear to you at your end, the bar, the long bar, the continuous bar, this is the heavy chain. And then the one at the smaller bar outside are the light chain. Okay? And this is the variable portion, the one in circle. This is where the antigen will bind or will attach. And then the one below is the constant region. And then they are bound by a disulfide bonds. There's 2 heavy chain, and then the heavy chain is bind to the light chain is bind to the heavy chain. Okay. So in an immunoglobulin molecule, if the light chain, there are 2 types of light chain, the alpha and the kappa, or the kappa and the lambda. Kappa and lambda man siguro to. Later, kalimot na ko, sorry. I only remember the heavy. Ang heavy chain, if If the heavy chain is a gamma, that's why it's called an IgG. If it's a mu, making IgM. So the name of the immune globulin molecule is because of the heavy chain. Apa ang lambda mo siguro to? Later we'll come across that. So, yeah. So the constant portion, this portion here, it determines the diffusivity of the antibody in the tissue, adherence of the antibody to specific structures within the tissue, then attachment to complement complex, and ease of passage through the membrane. So the light chain in a particular immune globulin molecule, permihe identical. So if the light chain is a kappa here, it should be a kappa also on the other, on the right side. If this one is a lambda, lambda po na dire. Likewise with the heavy chain. If this is a G, gamma, gamma na siya. So you cannot have a light chain lambda, the other one is kappa. You know? So identical. Okay. So next topic is so this one is the portion that attaches to the tissue, specific structure in the tissue, to the complement complex. I'm sorry. Okay. So what are the actions of the antibodies? So direct action of the antibodies on the invading agent, it can cause agglutination. So multiple large particles with the antigen on their surface bound into a clump. We call it agglutination. An example of this, it's very hard for you to imagine. You have seen how blood typing is done. It's an agglutination reaction. A drop of the blood, of your blood, and then they put that blue anti-sera and the yellow one. Or if there is no agglutination in the two, so you have type O. But if it clumps on the yellow and the blue anti-sera, you have type AB. So that is an agglutination reaction, the clumping. While another action of the antibodies is precipitation. So the molecular complex, your antigen-antibody complex is so large that it is rendered insoluble, so it precipitates, so precipitation. So again, it does not cause, if it precipitates so that for invading antigen cannot do anything, cannot do harm to you. Then neutralize. So antibodies can also cover the toxic sites of an antigenic agent and then neutralizes it at its action. And by the way, like say in a bacteria, particular bacilli, like say Salmonella typhi, it's not the whole Salmonella that is antigenic. So there are only certain parts of that organism, like say the flagella or encapsulated organism, the capsule. Okay. So what will the antibody do is neutralize that so that it cannot cause harm to the individual. And the last action is lysis. It's the antibodies, potent antibodies are occasionally capable of directly attacking the membrane of the agents and it causes rupture or lysis. Then another way is activation of the complement system. So what is the complement system? Although the complement system, we consider that part of the non-specific, but the antibodies can activate the complement system. So it's a collective term for a system of about 20 proteins, many of which are enzyme precursors. And they are, the principal actors in this system are 11 proteins designated as C1 to C9. So naging 20 kay upon activation, naging C1A, C1B, C2A, C3, C2B, etc. Okay, and plus B and D, factor B and D. Okay. So the next slide is on the complement system. Siguro ikalabad man ito. But I don't advise you to memorize C1, naging C1. So when I went when I was in med school, ganito ang pag-intindi ko, memorya ko, nako'y makalurong man ini. C1 to C1, ano, wala pa nga nini C1Q, na to. Ito naman din. Tapos, this one, so just understand, okay? So, the classical, the complement system can be activated 3 ways. So, surat rin ito wala din. So, by the classical pathway or the alternate pathway, and a third one by the lectin-Manan pathway. Okay? So, in the classical pathway, it is initiated by an antigen-antibody complex. So there should be an antigen-antibody complex to initiate or stimulate the complement system in the classical pathway. Okay. So what happens? A reactive site on the constant portion of the antibody molecule and binds directly with C1 molecule. Ito, C1 molecule on the system to start a cascade of activation. So what happens? Your antigen-antibody complex stimulates your C1. So you will have an activated C1, and then the activated C1 stimulates C4 and C2. You will have C42, activated C42, and then a C4A. So these are the product of an activated C42 and C4. Then this activated C42 will stimulate C3, and these are the products, C3b, C3a. Then C3b activates C5, so you will have C5b, C5a. C5b activates C6 and C7, you have C5, 6, 7. Activated C5, 6, 7 activates C8 and C9. And the ultimate product is C5b, 6, 7, 8, 9. Or the membrane attack complex, which cause lysis of the cell. So notice, itong naka-circle, circle, this in red, inside the red circle, these are the product of complement activation and their function or their actions. So an activated C3, it can cause opsonization of bacteria. We'll discuss that later. And then your C4A, your C3A and C5A, okay, 4, 3, 5A, or para madali, 3, 4, 5A, they will activate mast cells and basophils, and at the same time, C5A cause chemotaxis of blood cells. Diba? We said earlier in inflammation, the WBC are attracted towards the site of inflammation by a process known as chemotaxis. Okay. So this is C5A. And then the ultimate product is the membrane attack complex that will cause lysis of the cell. That's C5b678. So for you, just remember what causes the opsonization. So that's C3b. Tapos ito, activation of mast cells, basophils, C3, 4, 5A. Ito naman chemotaxis, C5A, and then lysis, C5b6, 7, 8, 9. What I told my students at Southwestern, ito, di ba ang mga jeepney sa Cebu, may mga 01K, 01A. Nasa ko, there's a jeep going to Cornelio, 01K. Amaldo kami memorya nga jeep, 01K. Would like to go to Colon, I take 01K. Tapos pagpabalik sa Western, take 01K lang nga po. No? Okay. Maglibog ka dito, may 01A. Going to may San Carlos area, nga kuan 01A. So 01K. Just like remembering which jeepney to take. Okay, kung lysis of the cell, C5b678 9. Okay. So, this cascade of reactions during the activation of the classical pathway. So, ito ang ano niya, ang mga product of complement activation. So, the other way of activating the complement is via the alternate pathway. So, this one, di rin na-start ha. You don't start at activating C1. So, start activating C3. So, microorganism plus factor B and D will stimulate. So, unlike in the classical, it's an antigen-antibody complex that will stimulate the classical pathway by activating C1, while in the alternate pathway, it's the microorganism combined with factor B and D, it will activate the alternate system. So the initial step is activating C3. Okay, so you activate C3, so you will have your C3 and down the cascade. Asya na. Okay. So effects of complement, opsonization and phagocytosis, so C3b. So opsonization is like it's a process of coating the antigen so that it will be more attractive, kumbaga mas matrasa siguro, attractive to the phagocytic cell. So it will cause the cells to engulf the bacteria to which antigen antibody complexes are attached. So that's opsonization. So I correlate this with kanang sa Ribisco na chocomalos, diba? There's a marshmallow coated with chocolate. Diba? If you just eat the marshmallow as is, puro marshmallow, wag ka nagkinaong gapas. But if you have that, the chocomalos na may chocolate coating, mas marasa ka unon ang marshmallow. So that's how I correlate that with opsonization. It makes the marshmallow more delicious, mas attractive. So yun, that's opsonization. It will coat the antigen, so it's easier for the phagocytes to mas siring pan kaupod ko. Lamian na si phagocyte makaon sa bacteria kay na-coat naman siya. Okay, then another effect is lysis. That's the ultimate, the latic complex C5b6789, or we call it the membrane attack complex. So it's a combination of complement factor and it has a direct effect of rupturing the cell membrane. Then another effect is agglutination. So your complement product change the surface of the organism, causing them to adhere to each other. And another is neutralization of viruses. Complement enzymes and other complement products can attack structures of some viruses and render them non-virulent. Then another is chemotaxis. So chemotaxis fragment C5a. Then activation of mast cells and basophils, fragment C3a, 4a, and 5a. So what will happen when the basophils and mast cells are activated? It releases histamine and heparin, and these substances increase local blood flow, increase leakage of fluid and plasma protein into tissues and other local tissue reaction that will help inactivate or immobilize the antigenic agent. Okay, so this factor play an important role in inflammation and allergy. Okay, maybe we can skip. So this is an activated complement protein. So what will they do? Destruction of invading microbes by the membrane attack complex. That's your C5b6789. Or it can cause vasodilation and increase capillary permeabilities, or chemotaxis, C5a ini, and then enhancement of phagocytosis, or by opsonization ini ang action inihan katong basophils, heparin na release by your basophils and mast cells activation. So the next is on the activation mechanism of activating a clone of lymphocyte. So it's different from the B cell, diba? Okay, each cell has a molecule similar to antibodies. We call that surface receptor. On the B cell, you have the immunoglobulin on the surface. This one, they have a surface receptor protein, or called a T cell marker on its membrane. And these also are highly specific for one particular or specific antigen. But the difference is The antigen that will activate the T cell must first be complex with our own body's cell membrane protein before it can combine with the T cell receptor. Okay, so this cell membrane protein, they are called major histocompatibility complex, or your MHC protein. So you just try to remember this mga abbreviation, because sometimes when I make exams, I'm lazy to type the whole thing. MHC, so MHC stands for major histocompatibility protein. So these are our cell membrane protein. So that antigen has to complex with this. And the host cell that carries the MHC protein are called antigen presenting cell. So macrophages, B lymphocytes, and dendritic cells, they all act as APCs, or antigen presenting cells. Okay. So special attributes of your T lymphocyte system, your activated T cells, and your cell-mediated. So upon exposure to an antigen as presented by macrophage, T lymphocytes will proliferate and releases whole activated T lymphocytes into the lymph. Your T lymphocyte memory cells are also formed. Diba? Upon initially activation of your T cells plus production of a memory T. Then T lymphocytes respond to antigen only when they are bound to specific molecules, we call MHC protein, found on the host cell. So on the surface of antigen-presenting cells in the lymphoid tissues. So what are these APCs? You have your macrophages, the neutrophils, and your dendritic cells. So the three major types. So ito na. Okay. Then interaction of adhesion, cell adhesion protein is critical in permitting the T cell to bind to antigen-presenting cells long enough to become activated. So MHC protein binds to peptide fragments of antigen proteins that are degraded inside antigen-presenting cells, and then they are transported to the cell surface. So there are two types of MHC protein. So MHC1, MHC2. MHC1 represents will present antigen to a cytotoxic T, while MHC2 will present antigen to a helper T. This one, remember, ang MHC1 cytotoxic, MHC2 present the antigen to a helper. So what's the role of the macrophage here? So most organisms, they are first phagocytized and partially digested by the macrophage. And the antigen products are liberated by the macrophage. Then the macrophage then passes antigen by a cell-to-cell contact directly to a lymphocyte, thus leading to activation of a specific lymphocyte clones. So in addition, the macrophage secretes specific activating substances that will promote further growth and reproduction of a specific lymphocyte, and this substance is called interleukin-1. So most antigens activate both T and B lymphocyte at the same time. So some of the T cells that are formed are called helper, secrete specific substances collectively known as lymphokines that activate specific B. So without aid of this helper T cell, the quantity of antibodies formed by the B lymphocyte is usually slight. So this is the so-called T and B cell cooperation. So it's this will stimulate your helper T, activate specific B to form antibodies. Role of interferon, as we discussed na natin eh. So the role of interferon, interferon is a cytokine, diba? In preventing, the role is it prevents viral replication. So most cell types, when infected with viruses, will secrete interferon. which enters the fluid and bind to the interferon receptors on the secreting cells themselves. So this is a virus-infected cell. It secretes interferon, and then it binds to the other neighboring cell. So it goes into the adjacent cell, and then some the paracrine function. In addition, some interferon enter the blood and binds interferon receptors on a far removed cells. Okay, so that's endocrine. This the binding, so itself misspelling. The binding interferon induces the synthesis of protein that inhibit viral replication should viruses enter the cell. So what happened in a virus-infected cell, it secretes interferon. Then it goes to the other neighboring cells so that these neighboring cells will not be infected by the virus. Or so in a paracrine manner or an endocrine. So endocrine kanang malayo pa nga cells. Okay. So what does it do is it prevent replication of the virus, but it does not. It cannot help itself. The virus, the infected cell, so infected na siya the virus. So even if it's producing interferon, it cannot help itself. So what it will do is it helps prevent the neighboring cells from getting infected by the virus. That's how interferon works. Let's go over this. Okay. Population of major group of T cells. So we have the helper T. The major regulator of virtually all immune functions. It secretes the following lymphokines: interleukin 2, 3, 4, 5, 6, and then granulocyte, monocyte colonies, ganglion factor, and interferon gamma. Okay, so helper T that are inactivated or destroyed by the HIV/AIDS infection. So it's the helper T that are inactivated. So in monitoring the progress of patient or making an initial diagnosis, you get the CD4 count. Okay. So these are the ones that are destroyed by the virus. Then another population are the cytotoxic and the suppressor. Specific regulatory function of helper T, stimulation of the growth and proliferation of your cytotoxic T, and suppressor T. It stimulates B cell growth, differentiation to plasma cell, and antibodies, and activation of the macrophage system, and it has a feedback stimulatory effect on the helper T cells themselves. That's why in the previous slide, I said that if your T cell, the helper T cell is necessary for the stimulation of the B cell, and it's the B cell that produces plasma. So kung waray ang helper T cells, you will have less antibodies formed. Your cytotoxic T is a direct attack cell that is capable of killing microorganisms and at times even some of the body's own cells. Okay. That's why there is a need for the suppressor cell or the regulator T cell. So for this reason, these cells are called killer cells. So after binding, your cytotoxic T forms a hole forming protein called perforins that literally punch around holes in the membrane of the attacked cell. So invaded by viruses, bacteria, or transplanted cells. So direct destruction of an invading cell by a sensitized cytotoxic T. Okay. So this is an attack, the cell, and this the dot at the surface are the antigen, patient's antigen. So this is a cytotoxic T cell trying to attack this antigen. While suppressor function serve the purpose of preventing your cytotoxic cells from causing excessive immune reaction So it explains immune tolerance. So tolerance of acquired immunity system to one's own tissue. So most tolerance results from clone selection during pre-processing. The failure of the tolerance mechanism causes the result into autoimmune diseases like rheumatic fever, glomerulonephritis, myasthenia gravis, lupus. Okay, these are autoimmune disorders. So what happens is there is a failure of the tolerance mechanism. So failure of the suppressor T cells. So artificial acquired immunity. Okay. So artificial acquired active artificial acquired immunity. So this is by injecting or in somebody with the antigen or okay. Well, we can have active, natural, acquired, like you have measles, sakit ka ng tipdas. So it's active, it's natural. Like, waray ka mo nito injecting measles virus. And then you develop an acquired immunity against measles. Hindi ka na magkaka-measles. So it's, okay, we are done by injecting dead organisms, like say in typhoid, in whooping cough, in diphtheria. So then, or a chemically treated toxin like in tetanus or botulism, or you can either be given an attenuated organism, attenuated live organism. Example is in the polio vaccine, the yellow fever vaccine, measles vaccine, smallpox, and many other viral diseases. While in passive acquired, you're not given the bacteria or a product of this organism. It's done by giving you the antibodies. So activated T cells or both obtained from the blood of someone else, from some animals or persons that has been actively immunized against the antigen. Like in COVID, di ba? You have that the plasma of patient who recovered from COVID, so they must have the antibodies to COVID. So they inject that into a COVID patient. It's a passive immunity, acquired immunity. Okay. So the antibodies last in the body of the recipient for 2 to 3 weeks, while activated T cells will last for a few weeks if it's transfused from another person, but it's a few hours only if it's transfused from an animal. So if it's from another person, lasting for a few days, well, from an animal, a few hours, a few days. This one is, you can give a lot of example like this, like in a newborn baby, the newborn has immunoglobulin G. By the way, maybe I don't know if I still have the many slides on the immunoglobulin. You try to read on the different types of immunoglobulin molecule, IgG, IgA, IgE, IgD, and IgM. Okay, so the 3 stages of specific immune response. Have you noticed I'm getting slower? Ikapoy na ko. Ikapoy na ko istorya. Sige lang, just bear with me. Matitimag hapit na nga. Yan ang pizza. So, initially, you have encounter with and recognition of the antigen by the lymphocyte. This is a specific response. Then it will be followed by activation of the lymphocyte. Then the next step is the attack, launch again by the activated lymphocytes and their corresponding secretions on that offending antigen. So yes, stage 1, during its development, its lymphocytes synthesize and inserts into a plasma membrane a single type of receptor. Tapos balik-balik na ini. That's why kinakapit niya pag binalik-balik. Pero para iyo. I hope this will sink in. Then if at a later time the lymphocyte ever encounters that same antigen, it becomes, you will have, the, because of the memory cell, so it's easier, faster and recognition. So as a result, the ability of the lymphocyte to distinguish one antigen from another is determined by the nature of this plasma membrane receptor. So each lymphocyte is specific for just one type of antigen. You remember that? Kaya nga specific. This, the progeny of the specific antigen-stimulated lymphocyte are called clones. And then it's estimated that in a typical person, the lymphocyte population expresses more than 100 million distinct antigen receptors, each with a potential to form a different clone. And the next step is the binding of the antigen to the receptor must occur for the activation of the lymphocyte. So upon binding, the lymphocyte undergoes cell division and the resulting daughter cells divide, and then the original binding antigen by a single lymphocyte specific for that antigen triggers multiple cycles of cell division. As a result, we call that clonal expansion. And then some of them become activated and the other become memory cells for future reserve for future encounter. So third stage is the activated effector lymphocytes will launch an attack against the antigen. Theoretically, it takes only one or two antigen molecule to initiate specific immune response. It will then result in an attack on all the other antigens of that specific kind in the body. So one group of lymphocytes, activated B cells, differentiate into plasma. So this is where the B and the T cell cooperation comes in. Okay, so your activated plasma secretes antibodies. Then another type of lymphocytes activates a cytotoxic T. The activated cytotoxic T, it will directly attack or kill the cells bearing that antigen. Okay, once the attack is successful, it's complete, great majority of the B cell, plasma, helper T, and cytotoxic that participate in that action, they die by apoptosis or the natural cell death. Okay, the timely death of these effector cells is a homeostatic response to maintain balance so that it will prevent the immune system from becoming excessive and possibly destroying our own tissue. If they do not die by the natural death, they will have an overwhelming number of these cells. And instead of becoming helper or helping us to defend the infection, they itself will attack our own tissue and cause disease. So, however, memory cells persist even after immune response has been successfully completed. So, matitiman na ini. Saan na ini nga slide? Matitiman na niya. So, we have this already in the first slide, so no need to go over them. Pinapoy na pagdinakan. Okay. So, it ends, so, right now. Tapos na. I think we're done. Siya. Okay, I think we're done with this. So, do you have any questions? No. Excuse ha, tawagan ko yung siya because I don't know how to operate this. Ako napakaon. Pwede parungot siya, masilaw. Sakit ang mata.
Dok. Human na, Dok.
Bumalik man yan, di dapat po pindot-pindot po.
Structure. Human na?
Nagawal pa pag, para wala ito, na masilaw. Bigla, ayun. Okay. Pero tuloy, kinakapo na, kaya na next week. Okay, I think we're done. Anyway, if I miss something, we'll still meet tomorrow. I still have 2 chapters with you. That's the chapter on blood typing, and then blood and hemostasis, blood clotting. Okay, so are there any concerns? How was it? How was the lecture? 10 now. Tomorrow daw at 10. Overwhelming. And actually, the topic, the module on immunology is not easy. Okay. Even I just get to like this because no choice. When I first started teaching here at RTR, and then when they transferred me to microbiology, no one would like to handle the immunomodule. So ako man maisog-isog. Maisog talaga. I tell you, tinodak ng maisog. Direct na ata sinaway. Pero direct makiaway yun. Wala ka mo kahadlok. Katapo mo ako downtown, di ko sasapakon. Direct, I do not start it away. Nagpupunay ko. Tinitikangan mula ako, di talaga ikaw tinuundangan. Okay. So, yun. By, wala man naruyag. So, I just had it. Then when I went for the postgrad, pag uli ko, ayun, kasi ako na. So, over the years, I just like it. Okay, there are advancement every year, may bago. Like, ngayon nga, there's COVID. You came across term like cytokine storm. So, yun. And it's very dynamic, ang immunology, the immune system. So, there's a lot to read. Madami ng article magbabasa. A lot to learn. So, kung at your end, second year, kumbaga, parang kasi padam nga, I like to do not stress yourself. At ako nito, when I was in the second year, nakukuha ko rin, ito pag-intindi. It was simpler din. Panla to, antigen, antibody reaction. Mga sugat lang ito. Korepat ng mga cytokine, ng mga interleukin, ng mga kuwan. Korepat naman una. Pero parang makuring nag-ihap intindihon at that time. Ang akong man attitude is, kung mag-exam baliman kung amo na ito, unless amo na ito i-exam kung quiz pero sabi ko, pag bi-monthly na, pero nalito ka question. I always set my goal as 100 na ito, kung mag-5 items yung immunology, 95 pa ako. Yun. Kasi, adi, marap kayo ni. Direct ka, mga pero nalito ka question. Anak 95, imanan 5, 90 pa ako. So, yung amo to attitude. So, yun. So, do not try overnight. Kalipong manadtong lecture ni Doc Aboy ba, immunology. Ay, lahat kaguol ay pagpay nakalurong para leto. Be sane. Kaya mo yan. As I said, I told you yesterday, try to read. First reading mo, wala ka kaintindi. Sige lang. So, basa otro. May ganat din magkukuha. Again, basa again. I had been nag-lecture na ako before I went to Malaysia for the postgrad in microbiology. Nag-teach na kato din, nag-lecture na nga kato immuno kay when the chairman din, Dok Sinisa, dahil ikaw na lecture sa kwan. Okay. Ako direct para kuan. Pero nakarawat dahil I complain later. Mapek nga kuan, mapek nga sundalo. I obey first and then I complain later. So yun. Then when I went to Malaysia, Kung iba naman nga immunology, mas complicated, manini, mas dami ma. So what I did was, I read. After the lecture, pag abot ka boarding house, I read. May binasa ako. I was trying to, kung magkarao din teacher ka na dito yung lugar, pag din yun, hindi ko maaram. So nagbibinasa ako, I read. There were three Filipinas in that group. Then, yun. So, the following day, nakaka-catch up ako what the teacher was talking because so, nagbasa ko nung kagabi, yun pala, that's how pala, sa platot karagsig nun. Parang ganon. Oo, but if you're really interested, magkaano ka. So, yun. Kasi yun lang ang ano ko, mahilig latak magbasa. So, you develop that habit because medicine is a dynamic science. Kadamot yung barasahon nga dati mo kaharap. Hindi ka makakaubos pagbasa. And I have that, the niya. Ang nga ni, I do not buy newspaper na. Okay. What happens if naubos, nakukonsume ko na puras, kaya na kuno na muna, pumalita ka newspaper, yung bayaran ko ito. So I read from the front page up to the last, pati obituary, pinanmasa ko na siya, uy, namatay na pala ikuan. Kasi nga na may classmate niya, kagrabe ni mo, dahil pati manak na ko, kaya ako nagipalit ang newspaper, sayang akong bayad. Basahon from pati advertisement, kasi ko, mga kuwan din may mga baligya, sa kanangkar, everything. So that's why, ko, di na kong paltog newspaper. Kaya, nakahurot na ang oras, sige, binasa ng newspaper, kaya kunto, nagbasa. So, yun. Para ang ako na nga, you develop that habit of reading. Sabi na yan nga, you just stay at home. Sige lang mo, stay at home. Ayaw ka mapagkinitat nga Korean. Okay na talaga sa manak ko. Nakikita ko, pero I get nasumo sa Korean. Pare-pare man na itong anaw nga Korean. I asked my granddaughter, Koy, ay nakita ito. Pare-pare man na ito mga anawong. I couldn't even remember. Hainit Jungkook, hainit Hirainat dita. Oo, nga Liminho. Kasi anak ako. Ani pa nito Liminho. I don't basta pareho lahat ng tsura tanang Korean. Nakumapay pat English kaya at least I can remember bisan diri ka magkitat na kay I watch TV and at the same time I do some light reading. Ako ay susiko nito, histalo nito nga boses or that's Liam Neeson. Boses na lang, di ka na lang kumikitat na wong. Oo. Nagot Korean kay parehas nga tanan it na wong. Yun. Pagbasa-basa ka mo. Especially those that you have a hard time in digesting everything, take time to read. Basa kay labi na you have a lot of time to stay at home and read. Hindi rin pwede makinadto Robinson. Dila ka mayubalay. So pagbasa-basa. Anyway, if you do not have the textbook, may mga e-book man ka mo to download nyo. Ako, I don't like that. Because pag open mo, like I had that in my iPad. Paspasin na kinarida. Tapos yun naman, ibatak ko yung iPad. And then, I have a hard time going through hain na ito nga pitch, hain na ito. But I prefer the textbook. So, if you do not have, you can come to school, borrow the book in the library. O pa nga niya na magbaro ka mong book, kulang tao nga din naka di library. But if you are in Tacloban, okay? So any concern nga ba ito natong coordinator dito?
Joanne po.
Joanne?
Yes po, okay.
So man, waray problema. I think, was I too fast?
You're okay po,
Okay
Doc.
la.
Thank you.
So if we did not, waray kita, concerned, anyway, tomorrow we'll have our last meeting. So we'll be discussing it was two short chapters. That's why we cut down the number of days for this unit 5. It used to be 5 days lecture, and una. Then I tried to cut it short. I used to chair the department, so I kanin ko kay Dok Palumay, cut na lang nato ni shorter kay kuan ganing kapoy nang hindi makaadlaw balik-balik sige, guan. So, yun, naging 3 days na. So, tomorrow, we have a short chapter. So, according to Misha, we'll meet at 10. So, if no concern for now, okay, thank you for your time, and I would like to see you tomorrow morning. Okay? Wala na,
Thank you,
Jo.
Doc.
Thank you, Doc.
You're welcome.
Thank you, Doc.
O sige, I'll ask Misha. I don't even
Thank
know
you,
how to
Doc.
turn this off. Ang matut akong problema, I have problems with any error internet connection. I don't even have, I have email. I don't even open an email because I don't know na why ano akong email, ano password. So if somebody asks me, do you have an email address? I have, but I do not use that. Alam mo na akong email address dito sa Southwestern. Ginimuan kami kanan Southwestern la. Ano to, su.edo.ph, etc. Then you have your own password. Wala kit makagamit because everything that is downloaded there, mga announcement, mga salary, everything. Sige la, nasulod man salary ATM man yan ito nga mga email. Kung sumulod ang ATM, karisigon ako nito. Okay. So sige, thank you and nice seeing you on screen.
Thank you, Doc. Merry Christmas.
Okay. So,